Hi, Aaron.
Hi. Hi, Renata.
Hi. Nice to meet
Good,
you.
how are you? Good to meet you too. How's everything?
Good, good. It's getting late here, but looking forward for it.
Yeah, great. Do you want to test out your slides?
Yes, I would like to do that. Let's see. Hmm? Hold on, I don't know why. Just a second.
Mhm.
It's not okay. Can you see the overall slides?
Yes.
I'm gonna do try to do PowerPoint presentation, see if that works.
Okay.
It's taking a minute, at least on my side.
Okay.
Can you see? How do you see it?
It's still just it's not the slideshow view.
Okay, slideshow. Let's see if that works. Does this work?
Yeah, that looks good.
Perfect. Okay, great. So 20 minutes plus 10 for questions, right?
Yeah, yeah.
And am I going first?
Yes.
Great. Thank you.
And I'll say a little bit of introductory thing and then you'll go.
Great. Let's just skip through them. You can keep seeing them, right?
Yep, looks
Video
good.
is working. Okay.
Mhm. And then if it looks fine to you, you could stop sharing for now.
Okay. Hopefully it will work again once. Let me maybe try one more time. Should be fine.
It's always good to be sure.
Okay, let me just one more time just to make sure. Share. Can you see it good?
Great. Yep.
Okay, great.
Yeah, thanks for doing this.
Of course, my pleasure. Thanks for inviting me. I appreciate it.
Mhm. How long have you been at Einstein?
It's been 4 years, exactly 4 years. But
Oh, great.
yeah, but you know, two of them have been pandemic, so it's been.
Mm,
It's just like my lab was just like really starting to take off, and then we were closed for over a year, and we lost our almost all of our mouse colonies. So it's been
oh no.
rough,
Do
but.
do they like take that into consideration and give you more time or help you
You
or
know,
something?
not yet, but I'm trying to negotiate that just because, you know, it's
Yeah.
like we were forced, we were not assured minimal services. We were forced to put down all the mouse code in the end. All my work involves various genetic lines. So it just took a year to build a colony, and then we were down
Mm,
for 2 years, and now another year to rebuild the colony again. So
I'm sorry. Yeah. It like
Sorry to hear
definitely
that.
hey, so
Renata.
hi.
now
Renata.
now it seems like all research is stopped in Shanghai, right? So
Yeah, I'm afraid the same thing happened with everything stop. So luckily we have some personnel in the mosque facility to take care of them. So to barely maintain everything going on there. So we don't have to put down the colonies. Otherwise, it definitely take years to cover.
Yeah, it's taking years for me for sure. And
Yeah.
I mean, honestly, it's still like I still haven't recovered more than half of my lines. It's really brutal.
Yeah.
so I mean different universities did it differently, but at Stanford they said you can't work in a lab unless it was COVID project. So all of my projects became a COVID project and we just kept working, not on COVID, but we just kept working on our normal stuff. We had to do shifts throughout the night, but we didn't stop for a single day.
Yeah, I mean, for us, nobody could access the building. I mean, there was not even minimum services assured to maintain the animals. Leave out to go to the lab and do any kind of work.
Oh, Wow. No, we had to I had to write all these things saying like why it was an emergency and stuff, but I don't think we really stopped working for a single day.
Yeah, I mean, so I think that different labs really got affected differentially.
Yeah, for sure. And different people at different stages. Like I think
Yeah,
it really would affect you because you had just gotten your lab rolling.
exactly. I mean, it's like there was like you couldn't analyze data, or like there was there was nothing. But
Yeah.
also, we are catching up now.
Yeah. Good. Good.
Yeah, Gang is working on his Zoom version. Yeah.
Okay. Hi, Jin.
Hi.
Hi, Jin.
How are you guys?
Good.
Hanging in.
Yeah.
Okay.
Still has some hope.
Good. It's
No,
always
I don't
good.
have hope anymore. I had hope last week.
What happened meanwhile?
So I'm living in
Okay.
Shanghai this year, and there's a full city lockdown because of COVID.
Oh yeah. I didn't realize you were living in Shanghai. I get it.
Yeah. On a sabbatical.
Well, the May 1st holiday is not gonna happen, right?
I think they're still off from work.
Well, so meaning next week. Usually
Yeah.
there's a May 1st holiday. So probably everybody works harder. Aaron's not to make you feel bad. And I'm showing a Zoom background on the West Coast.
Oh yeah, I see the hang gliders. Yeah. I booked my plane ticket to California. That makes me feel good.
Oh, good.
I don't know how I'm going to get to the airport or how I'm going to leave my apartment, but at least I have an airplane ticket.
Yeah, months will be fine. Yeah, I believe certainly.
But they told us 5 days, right? They said 5 days Pudong, 5 days Puxi, and it's been a month.
Yeah.
Can you just wear those safety suit to get out?
Good idea. So I was thinking of wearing one of those and blending into the crowd.
Well, you don't want to do that. That is against that will get you in trouble. But you can leave heading to the airport.
Yeah. But it's a little bit tricky because to get into the airport, you need to have 2 COVID tests, but it's hard you have to leave your apartment to get those COVID tests, which is hard.
Oh.
It's not impossible, but it's complicated.
Just still check the regular, right? Every other day or something.
Yeah, but I think you don't need one of those group tests. You have to get like a individual one.
No,
the pool
you
test.
I don't have the good test. You had the asset on your phone. You should, yeah, you're right. That's some improvement to show that.
And then
Yeah,
I
Gang's
heard
still working on that. Mhm.
Okay, I heard so from my apartment to the airport, it's like 30 minutes, usually 200 renminbi, which is
Yeah.
about $35. But now it's like 12,000 renminbi because it's like
Good.
which is like $2,000.
Wow.
Some
Yeah.
black market.
Yeah.
Is that expensive?
Around. Well, I heard that, but yeah.
Oh.
Well. Oh, hi, Li Hui.
Hi, Aaron. Hi, Renata.
Hi, how are you doing? Nice to see you again.
Nice to see you again. Yeah.
All right, if Li Hui is here, it means we can get started, right? So.
Why not?
Okay, cool. All right, welcome everyone. Another Shanghai lockdown edition of NeuroZoom. Hope everyone has enough food, but we can provide the science, and we have great talks. Gang is on his way, but Renata will start anyway. But next week, please tune in. We have 2 more good talks. We have Perry Kirschan. So a theme of NeuroZoom is actually we have speakers, and then we even have the former trainees of their speakers. So Perry Kirschan was a trainee of Kang Shen, and Renata today was a grad student with Gord Fischel. And then we have Julia TCW, I think the only NeuroZoom speaker whose last name doesn't have any vowels. So that'll be a great speaker. She's a great scientist, has made major discoveries about ApoE and Alzheimer's, and she'll be coming next week. So with that, I'd like to introduce our first speaker today, Dr. Renata Batista-Brito. She's an assistant professor at Albert Einstein College of Medicine. She's originally from Portugal. Then she did a master's degree in Paris before returning to Portugal for her PhD. But in Portugal, she in our PhD program, she joined a lab in the US, and she went to NYU to study with Gord Fischel. And here she started working on cortical interneuron development. And she discovered an important role for transcription factor SOC6 in the function of interneurons, and she showed that it functions in a similar way to LHX6 and actually functions downstream of LHX6. And she showed that the mice that have impairments in SOC6 have some epilepsy. She continued to work on interneurons for her postdoc when she went to Yale with Dr. Jess Carden, but here she moved on from sort of genetic analysis of interneurons and now also worked on how interneurons integrated and functioned in circuits in the visual cortex. And here she used mouse studies to study neuregulin ERB4 signaling and the role of VIP neurons in circuit integration and development of cortical circuits. So with that, she's been for about 4 years at Albert Einstein in her own lab, continuing to study interneurons and circuits and their role in sensory perception. And we'd like to hear your latest. Please go ahead, Renata.
Well, thank you so much for inviting me and for the kind introduction. And I am very excited to share the work that has been going in my lab for the last couple of years. This is totally unpublished, still preliminary. It was, all this data started just before COVID. And then we were closed for, almost this whole time and lost our mouse colony. So it's an ongoing process, and I really welcome feedback on the project as much as possible. So before anything else, to do credit, so most of what I'm going to show you today is the work of a very, very talented PhD student, Jacob Ratliff. And so my lab at Einstein is really interested in sort of 2 main thematics. One is how the brain processes information according to the behavior state in which we are in. And we all know that, for instance, 2 extremes would be like when we are asleep versus when we are awake, you're going to be processing information totally differently. But also if we are awake and if we are engaged and paying attention versus if we are awake and drowsy and sort of dozing off, the way that we are going to, for instance, perceive this talk and what I'm going to tell you is going to be very different in the way that you're going to remember it later, what you're going to understand, and etc. And so my lab is really interested to understand what are the circuits that mediate this sort of state regulation and how they impact on information processing. And one of the particular sort of fields within these circuits that we are interested in is in cortical inhibition. And that spears from my work at the Gord Fischel lab. And I'm pretty confident that everybody here knows that inhibitory neurons come in a lot of variety of flavors. They are very diverse in terms of their intrinsic properties, the markers they express, how they connect. They are unified by the fact that they are local, so they project within the local region, and they are inhibitory usually through the expression of GABA. And so here on the right, you see, a beautiful Causal drawing for illustrating some of the different inhibitory classes in the cortex. So for the rest of the talk, I'm going to show a subset of data that is going to be focused on in vivo extracellular recordings that we are doing in mice, such as the one that you see here. While we are recording local field potentials and doing basically extracellular recordings with liminal probes while the mice is awake. And these mice, we are monitoring their state by looking at pupil, whisking, locomotor activity, EMG, EEG. And with these linear probes, we can record local field potentials that, basically is the sum synaptic activity of the network, as well as a variety of excitatory and inhibitory neurons simultaneously. And then we analyze how the sort of activity in different brain areas, but we are going to focus for this talk in the visual cortex, changes as the animal moves from, quiet to, for instance, alertness. And I told you that we are going to focus in inhibitory neurons. And one of the problems that inhibitory neurons have been having in terms of address their function in the network is the wide variety that they are. And so we have been using in mouse genetics to try to nail what is the function of different classes of interneurons. And you saw probably a talk by Gord recently showing sort of like different neurotypes do different things, have different origins. But the main caveat that has been having in all these studies, including in VIP interneurons in my postdoc, is that each class of interneurons that we labeled with Cree lines or flip lines in mice, such as somatostatin or parvalbumin, encompass a huge variety of subtypes of interneurons. So each time we are modulating one, what we say to be a neuronal type is actually really a variety of neuronal types. And they might have different projections, they might, talk with cells in different ways, and they actually might have sometimes opposing effects. And when we address them from the perspective of circuits, it becomes really, really complicated. And that is something that was a struggle for me in the past and that I really wanted to address head on in my lab. And so when we started to think about, how to go after subpopulations of neurons that are actually kind of like clean in terms of their function, we start to look first at the transcriptomics. And here I'm showing you a TSE plot for that is illustrating the transcriptomics of different classes of interneurons or inhibitory neurons, GABAergic neurons. And you can see like the candidates somatostatin, VIP, PV, ID3, which are the ones, the neurons that come from the CG. And then in the midst of all of these, you did this little island here of a subtype of inhibitory neurons that express somatostatin and nNOS, and I'm going to refer to those as somatostatin and nNOS cells. Now, this is a very, very small island, which shows that it's a very small subpopulation of inhibitory neurons, but it's also an island that is very isolated for all the other classes. So they are quite unique. So their uniqueness call my attention, but also the fact that despite being so low in number, they are the most conserved neuron type across both excitatory and inhibitory neurons, all the way from amphibians to humans. And they expand evolutionary, being that humans have the highest numbers relatively, to other species. So when we start to zoom into this, we saw in the literature that this inhibitory neuron is even further more, strange than other inhibitory neurons in the sense that they are long-range projection neurons, right? So they are inhibitory neurons in the neocortex that project long range. The full extension of their projections is not known and is part of what we are trying to address. Their inputs is not known, is part of what we are trying to address, but for sake of time, I'm not going to address these points in this talk, and I'm going to talk about their function. So this is just a quick video to say that, so we are going to try to address the function of these neurons in vivo recordings. And here I'm just showing you an example of a recording in which we are measuring local field potentials. You're hearing one cell, and as the animal is still, and you're going to see that, as we all intuitively know, as the animal transitions state, and in this case, as it's going to start locomoting, anticipation of locomotion, its pupil is going to open. You can see the network becomes really desynchronized or dominated by high frequency oscillations. In this case, you hear a cell really firing, and here you can see the trading of moving. So obviously, as the animal is transition state, the network is really, really different. So we had some hypotheses that these somatostatin and nNOS cells might be important for state dependence modulation of cortical networks, but our big struggle is like how to look into these cells, how to target them. So we use a strategic intersectional approach So these cells, these long-range inhibitory neurons, have to be labeled by the intersect of two different genes, somatostatin and nNOS. So we are crossing somatostatin flip mice and NOS pre-R mice, and then we use intersector viral vectors that only express, whatever downstream protein at the intersect of the expression of pre and flip. So we use the intersect viral vectors for various different types of manipulations. And here you can see that if we do that, we selectively and exclusively and quite reliably label the somatostatin nNOS cells that, as you can see, are very sparse in terms of the number of their numbers in the visual cortex. Now, what we know about these somatostatin nNOS cells? So we know they are long range. We know that they express somatostatin and nNOS, two powerful neuromodulators, and so through their outputs, they release GABA, NPY, somatostatin, and nitrous oxide. In terms of their inputs, we don't know much, and we are exploring it further, but we know that they receive strong inhibitory inputs of acetylcholine through M2, M4 receptors, and also inhibitory serotonin receptors, as well as GABA. And the excitatory inputs are thought to be mediated by substance P and glutamate. And this is still a little bit of a topic of polemic discussion. In terms of what these cells might be doing as per their function, the only clue that was really in the literature at the time was that they might be involved in sleep. And that study that we are showing here by the Kidolf lab shows that if you take a mouse and you sack it immediately after sleeping and you stain for C4 cells, some of the cells that express higher number of C4s are these somatostatin nNOS cells, suggesting that they might be active during sleep. So with this, we went on testing our hypothesis that during the alert state, these cells are receiving inhibitory inputs mediated by, for instance, inhibitory inputs from acetylcholine and are inactive, and that allows the network to be in a desynchronized state while the pupil is big and locomotion might occur, and that the activation of somatostatin nNOS cells would appear during quiet state and sleep, and that would lead the network to be synchronized. And that's what we are going to test next. And so the first thing we needed to try, we decided to do, was to see what is the natural activity of these long-range somatostatin and nNOS inhibitory neurons in a mouse in vivo. And to do that, we devised sort of this preparation whereby we are doing calcium imaging of somatostatin nNOS cells positive. We are also going to be doing calcium imaging of somatostatin positive nNOS negative cells, the other somatostatin cells that are not the long-range inhibitory neurons in terms of comparison. And we are at the same time doing EEGs, EMGs, and LFP recordings, both in the hippocampus and in the visual cortex, in order to monitor both network activity, whether it's firing rate of other cells, as well as local field potentials, and monitor their states, such as sleep and different states of wakefulness, and also different states of REM sleep and non-REM sleep. And so this is an example of such an experiment. So here we are using intersectional genetics to both label all the somatostatin cells with GChem, and then we are labeling the nNOS cells just with mCherry, and by using this intersectional dual color imaging approach, we can know whether we are imaging for somatostatin cells only or somatostatin nNOS. And then we image them along locomotion, facial movement, to have an external measure of arousement, as well as EMG and local field potentials. This one is in the visual cortex, and we also do it in the hippocampus. And what it became clear once we start doing these recordings is that somatostatin nNOS cells are active when the animal is quiet. Here I'm not scoring for sleep, but it's both in transition into sleepness and when they are asleep. But when the animal is really sort of like in the drowsy or quiet state, and never when it is locomoting or whisking or with high pupil. That pattern of activity that you can see here of periods of somatostatin nNOS activity never is sine qua non with high arousal. In fact, it's low arousal. It's sort of different for the pattern of activity of other somatostatin cells, which tend to be more active in periods of high arousal. And this activity is really correlated with low frequency oscillations in 1 to 10 hertz, really like in the delta sort of 1 to 4 hertz frequency. We went and we quantify these metrics. And so here what you're seeing is the measure, so the video energy for phase motion, EMG, and locomotion for periods in which somatostel cells are inactive or active. And you can see that they are active. There's like mouse is not moving their face, EMG is low, is not locomoting. Sorry. And you can see that if you do the power difference between active and inactive, the cells are really, you can see that they are really firing during the period of low frequency oscillations. On the contrary, when we do the same metrics in the somatostatin positive and nNOS negative cells, those would be the canonical Martin-Arty somatostatin cells, as well as other sort of local somatostatin, more typical inhibitory neurons, we see the opposite trend. So they tend to be active during periods of higher arousal, and actually during periods in which their activity is on, there's suppression of low frequency activity. Okay, so we saw this correlation between low states of arousal and low frequency oscillations and the activity of these somatostatin nNOS cells. And then we wanted to ask if there's a causal relation between these low frequency oscillations and the activity of nNOS cells. And to do that, we did 2 experiments whereby we are either gonna express chenorodopsin in somatostatin nNOS cells, or we are gonna express chenorodopsin in the other somatostatin cells. And then we are gonna activate either the long-range somatostatin cells or the other somatostatin cells with a variety of protocols, while performing extracellular recordings as before and measuring all the measures of arousal as before. And when we do that, what we see is that, for instance, if we do a white noise optostimulation of somatostatin nNOS cells, and we look at the transfer function, at the gain of the transfer function, we see that the activity driving somatostatin nNOS cells with white noise really increases the gain in these low frequency oscillations, suggesting that they might be actually inducing these oscillations. When we then do a variety of different protocols, both with square pulses or sinusoids of different frequency, what we consistently see is that the activity of somatostatin nNOS cells drives low frequency oscillations in, like, as I said, like 1 hertz to like 4 hertz activity in the delta range. That is significant, is robust, and it happens no matter what kind of protocol we give, whether it's like a square pulse or different types of sinusoids. And surprisingly, we can induce these patterns of activity not just when the animal is still, so we not only increase sort of like low frequency oscillations when they are sort of still, but still awake, right? But even if they are in movement, which is something that is, certainly the opposite of the network being in low frequency oscillations. Now, when we do the reverse experiment and we, instead of activating somatostatin nNOS cells, activate the somatostatin cells that do not express nNOS, we see the opposite. So we see a suppression of low frequency oscillations. So we wanted to know if this effect is like a local, because here in this experiment, we are just injecting the visual cortex and just activating somatostatin nNOS inhibitory neurons in the visual cortex. And we wanted to know whether you can see the propagation of the effect to faraway distances. And right now we actually are doing sort of linear probes across long distances and look at the decay over time. But even if we put an electrode in the contralateral V1, so like on the other hemisphere of the brain, we still see an increase of low frequency oscillations when we activate somatostatin nNOS cells. That's not as robust as when we do like in the local side where we inject the virus, but it's still significant and much higher than if we don't induce it. So these data taken together really suggest that somatostatin nNOS cells are increasing the synchronous network activity of the local network, as well as over long distances. And we wanted to go further and see, outside of the local field potential and look at neurons themselves and see if there's an increase in the spike synchrony of neurons recorded when we activate these somatostatin nNOS cells. So here we are doing, I'm showing a spike synchrony plot in which we are looking at cells fire together during non-optostimulation and optostimulation. You can see that driving somatostatin nNOS cells increases the synchrony of other neurons. In this case, the vast majority are excitatory neurons. And when we also look at phase locking, or like entrainment of the network to different frequencies when we activate these cells. So again, so here we were seeing, like, when we are talking about this population, we are like maybe having a handful of cells. We are, we are exciting 10 cells, 20 cells, really a small amount of cells. So I really wasn't actually not expecting that we would see anything in terms of network phase locking or network synchrony. But we can see very clearly that during the optostimulation, we see an increase in network entrainment in the 3 to 6 hertz. And you can see it measured here as BBC. So we also looked at what are called down states, or like basically suppression of firing activity, akin to some down states as has been measured in vivo by others. To do that, we look at the power. So we are trying to figure out whether neurons, that the network has more or less down states when we activate somatostatin nNOS cells. To quantify down states, we use this metric that is suppression of gamma power and silencing in the multi-unit firing activity. And so when we look at the number of down states in the non-optostimulated in gray, or optostimulated in blue, or of somatostatin cells, or optostimulated of other cells that are not somatostatin, and we score our data in awake and sleep, what we see is that both during awake and during sleep, somatostatin nNOS cells really drives a huge increase in the number of down states. That is despite that the average firing rate overall, during the recording time is not changing. So it's not that you just have like a overall suppression of the firing activity of cells, and therefore you see increased down states. It's the temporal dynamics that change. So even though the overall population firing rate doesn't change, you really see this massive increase of down states during sleep, and something akin to down states. Down states is really a nomenclature just used for sleep, but basically suppression of firing in awake. So to conclude, I just show you that we study this new cell type, or new, not under discussed and represented cell type, that is an inhibitory cell type. That is very weird. It's a long-range inhibitory cell that expresses GABA, and that we show that it's active selectively during the quiet state. Now we actually have recordings scoring different epochs of sleep, and they are really active during slow wave sleep, but totally suppressed, non-active during REM sleep. And that is during this quiet state, they are dominated by periods of synchronized activity. And during the alert state, we show with the calcium imaging that these cells are inhibited. We are now doing the pharmacology to try to address, the mechanism by which they are excited or inhibited. And during this alert state, the network is asynchronized, possibly in part due to the suppression of the activity of somatostatin nNOS cells. And then I also show you that driving the activity of somatostatin nNOS cells is sufficient to synchronize the network to these low frequency oscillations independent of the state of the animal, and that this sort of like network entrainment to low frequency oscillations can be felt in the place where we are activating the cells, or even over long distances. And so that's it. I just want to thank my lab and our collaborators. So this work was fully done by Jacob. but we have been having the pleasure to collaborate with a bunch of great colleagues. We collaborate with the Schulzen lab. We sort of have a logistic shared lab where we share techniques and some rigs and some expertise. With the Tomer lab, which I didn't show you, but with whom we are collaborating to sort of analyze the full extent of the output of these cells by doing single cell reconstruction in cleared brains. With the Dythruth lab and the Fino lab, that has provided some of the tools that we used. And I would like to thank for funding, for our funding institutions.
Thanks so much, Renata. Awesome talk, and we're open for questions. Kaiwen, of course.
Hi, a great talk. And so I have a couple questions regarding this very unique type of SST neurons. So I might miss some of the points. And did you mention that you did the recording in a specific cortical regions, or you think these properties of this SST neurons is universal across different cortical modalities? And the second question is, and I kind of remember that you mentioned that you stimulate SST, this SST nNOS neurons, and you change the power, the slow wave power in the contralateral cortex. So have you monitored that whether manipulating the activity of this type of neuron will change the quality of the duration of sleep or any property of the slow wave sleep? Thank you.
Okay, so yes, great question. So first question, where did we record? And we think that something specific about this region or is a generalized property. So we recorded in the visual cortex, in the two areas of the visual cortex. And so the data that I show is in the visual cortex. I do not, and so we are also trying to understand how these cells might be influencing visual processing. But I do not think that the activity of the cells is specific of the visual cortex. It's just the place that is convenient. We are also now recording from different areas, and I don't have the data to show you, but I think it's going to be pretty general across the cortical areas. So as per the second question, so what happens so I think you want a confirmation whether the activity of these cells increase low frequency oscillations, and that's correct. So that's I showed that. So and then you were wondering how does it might affect sleep, right?
Yes.
And so that's a question. So that is like a really difficult experiment for us, and that's because all our tools rely on viral expression. And so what we are trying to do right now is to activate and suppress them, but in the whole cortex. So we are doing Swiss cheese mice, where we have injection all over the cortex of the virus. And so that we have a general manipulation that is just in the neocortex, but broad enough that, might allow us to figure out if either activating the cells drives the animal to sleep, or suppressing the cells drive the animal to not sleep, right? So we are doing those experiments right now, but it's really challenging. We are, the other, in terms of sleep, the other thing that I can say is that there's the activity is highly correlated with sleep. So when we do calcium imaging and we look at slow wave sleep and we look at REM sleep, it's obviously that the cells start to be active as the animal is still awake but drowsy, right? Sort of like slowing down. And then if the animal wakes up and twitches, the cells immediately silence, sharp. But then if they go into sleep, their activity really ramps, becomes super high. And then if they transition to REM sleep, it's like a vertiginous precipitation. Mm-hmm. So that's correlation. Whether it's causal or not, we are doing the experiment that I just told you.
Okay, great. Thank you.
Thank you.
Okay, Shu.
Hi, Renata. Very nice talk. Yeah, I have a very small question. So for this induced downstate, do you think they will propagate to other area where you do not stimulate the cells?
Yeah. So So what I can tell is that I think they do propagate, but the difficulty that I'm having, so the caveat to that is whether it's like a direct connection or indirect, right? So you can imagine that's like, is it because of this direct connection or not, right? So it's because like basically the network propagates. So and you induce like sort of like low frequency oscillations and down states that propagate in the network through excitation, not necessarily through inhibition, or is a direct product of the inhibition. So that's the difficulty. So to address that to the more precise temporal dynamic, what we are doing now, we are doing custom-made probes in which we are actually recording a long distance, but in close by proximity. That's one technique. And the other thing that we are doing is that we actually are building a wide field microscope akin to a mesoscope to see if we see the propagation of these waves as we activate them, over time and see, if we silence those cells, if they no longer propagate. But they do propagate over long distance, but I cannot tell you it's like a direct product of the inhibition. It can be a side effect of reduced excitation.
Okay, thank you. So also, do you know where these cells project to?
Yeah, so we are looking into that closely. So they project really, really widely. So they, as far as we can tell, they really project only within the neocortex, even though we are like, this is ongoing. So we are cutting many brains and look at the extent of their projection. But it's so far, at least we only saw them projecting in the neocortex, and they project, like we are, let's say we are labeling in the visual cortex that project all the way to prefrontal areas and all the way to the most extreme back of the cortex, and they project also ipsilateral to the same region. So if they are in the V1, they project to the other V1, right? Ipsilaterally. So this we know from looking at the population of cells, but now what we are trying to figure out is actually to do single cell reconstruction in cleared brains and look at the full morphology and ask, do they send collaterals? Let's say they project to all the way to the prefrontal cortex. Do they send collaterals all the way, or do they just project to the terminal? Do they project to specific layers? Are cell bodies in certain layers particularly projecting to another layer? So all of these things have to be done with a not population labeling, but single cell labeling, and we are working on that.
All right, thank you so much.
My pleasure.
Li Hui?
Hi, thank you. So great talk. I just have a very quick question. So since these cells seem to be more active in a quiet state, I wonder whether they are modulated by neuromodulators such as acetylcholine or norepinephrine.
Yes, absolutely. They are suppressed. So they are the cells in the cortex that express the highest level of the M4 receptor for acetylcholine, which is the inhibitory receptor. So it's like extremely high levels. They also express inhibitory receptors for serotonin, but they also express like receptors that are thought to be involved in sleep, hypocretin and things like that. So right now there's like really nothing known about the pharmacology of the cells except like the single cell sequencing data that
right.
So now we are actually trying to do both. We are starting to do some slice electrophysiology and some in vivo pharmacology to have a better idea of what are the neuromodulators that are driving them. But your suggestions are totally on target, I believe.
Thank you.
Thank you.
David, do you want to ask your question?
Yeah, I just have a quick question. Do you have any insights onto the spatial distribution of the boutons on these axons?
We don't, not yet. So would be great to know. So we are also, so this is like, because there's like so little unknown, it's like I have to tackle it from all these angles. So we are right now, we just did some processing of tissue for electron microscopy to try to figure it out. So I don't have the answer, but we developed the tools. We did some gold staining. It seems that it's working, and now the idea is to look at the sort of the microstructure. But for that, I think we need to go a little bit past just the light fluorescence and go into electron microscopy.
Okay, awesome talk. There are 3 questions in the chat. If you could, while we go to the next speaker, if you could answer those are very good questions. Thanks for sharing your great study. Just type the answers in there as you go. And then DeLong, is Gang ready?
Yeah.
Should I just type it, right?
Yeah, just type it. Yeah.
The chat bar. Okay. Thanks, Netta. Great, beautiful talk. So it's my great pleasure to introduce the Dr. Gang Cao, who is a good friend of mine. So Gang took a long detour towards neuroscience. So Gang has got his bachelor degree, a master's degree in Chinese Academy of Sciences. After that, he went to Netherlands, did a PhD with Nijmegen University, and after PhD, he went to States. Did the first postdoc with Stuart Lipton in Stanford Brigham in San Diego. After a couple of years, he went to did postdoc with Tony Zetter in Cold Spring Harbor, where he started to use all kinds of various of viral tool to dissect neurocircuitry, which is some kind of complicated. I couldn't understand for now. So after 2013, so Gang come back to China to start his own lab in Huazhong Agricultural University, where he started to use all kinds of viral tools to dissecting interaction between the neural system and immune system. I believe some of them he will talk about today. Okay, welcome, Gang. Take it away. We can see a slide. Great.
Yeah. Okay. Thank you. Thank you very much. I'm sorry for this situation. I was I changed 3 computers and finally logged on. Thanks for the invitation, and it's my great honor to join this great work seminar. So today I would like to give update about our recent published paper about neuron-derived neuropeptide, why fine-tune the immune response. So, okay. Okay, so this is the outline of my talk. So firstly, I would like to give some introduction. So a long time ago, the nervous system and the immune system were thought to be independently operated. But a few decades ago, people get more and more evidence that the nervous system and the immune system were closely associated with each other because they share the same neurotransmitter and cytokine receptors. So that means the nervous system express more or less all the cytokine receptors, and the immune systems express more or less all the neurotransmitter receptors, including the excitatory neurotransmitter receptors and the inhibitory neurotransmitter receptors. That means they both can share the more or less the same language. And recently, more and more people working on this called neuroimmunology field. Especially the regulation of immune system by nervous system. And recently, people even think that the cancer can be also the progression and of the cancer can be also regulated by the nervous system. Also, people found that the nervous system can sense the invasion of pathogens and dynamically regulate the immune system balance. And then, but still, there are a lot of knowledge is unknown in this field, and people. Most of our organs are connected with the nervous system. For example, the biggest immune organ, the spleen, they are highly connected with our nervous system. As you can see here, I will use my lens. Okay, as you can see here, the spleen are highly connected with nervous system. That means they should be elegantly regulated by the nervous system. Okay, so but how the nervous system communicated with the immune system? So there are basically there are 5 pathways to regulate the brain regulated immune system. We have 5 pathways. For example, the endocrine pathways, the sympathetic nervous system pathway, and the sensory pathways. But the people know that the lot of transmitters can released from nervous system and then regulate the immune system. So, for example, the NPY neurotransmitters can hypothesis to involved in the regulation of the immune system, but the detailed molecular mechanism are largely unknown. So if recently people proposed that if we can apply the nervous system regulation on immune systems, so we can do a lot of clinical application. For example, they can use the electrophysiology stimulation of nervous system to cure the autoimmune disease, pain therapy, and cancer. And also people think they can be used for diabetes and hypertension. During last 2 years, Professor Qiu Ma published 2 important papers and illustrated that the mechanism of acupuncture. And they proposed that NPY positive neurons are involved in this process. So today I would like to give introduction of our study. So we are in our group. We are working on the system, the neural system biology, about the interaction between nervous system and the immune system, and how the neural system sense the invasion of a pathogen and then regulate the immune system for virus clearance. So we found that upon infection of the pathogens, the ganglia, the celiac ganglia can be activated by this by the infection of pathogens. As you can see here, the C4s are highly expressed in Sage ganglia, and then we found that the NPY are most significantly regulated neurotransmitter related genes, as you can see here in the. Okay, I will come back to this one later. But the take-home message of this slide is that the LPS can activate the Sage neurons and upregulate C4 expression. We also performed the immunohistochemistry and found that the LPS receptor, Toll-like 4 receptors, are highly expressed in this ganglia, and they are more or less co-localized with the retrograde labeled neuron signal, the PRV virus signal. So that means the Toll-like 4 receptor positive neuron projected to spleen, and if we treat them, if we treat these neurons with LPS, the neurons can be highly activated, as you can see by the calcium imaging analyzed here. That means the Sage neurons can sense the invasion of pathogens. As I mentioned in the first slides of our work, we found that the Sage neuron, particularly the NPY positive neuron, they can respond to the inflammation or pathogen invasion, and then significantly significantly upregulated the NPY genes. We also can check other neurotransmitter related genes. For example, the NE related genes, GABA, acetylcholine. The genes are not upregulated in comparison to NPY genes. We also use another ganglia in SG, which can regulate our heart. We found that in this ganglia, NPY are not regulated upon the RPS treatment. But you can see the NE related genes are slightly upregulated, but not NPY. That means NPY, the dramatic upregulation of NPY is specific in CG neurons. We also performed immunostunning and demonstrated that the NPY neurons are co-localized. The NPY neurons are co-localized with the presynaptic protein synaptophin in spleen. Suggests that the NPY can be released from this vehicle. So this picture were taken in spleen. That shows the NPY are co-localized with a presynaptic marker. That means the NPY can release from the nervous system to the immune system. Next, we use AAV siRNA and specifically downregulated the CG neuron NPY. So we inject AAV si NPY in CG, and we tried two different the type of siRNA and AAV virus, and we showed that indeed the NPY are downregulated in CG neurons, and we showed that after this lockdown, we can see we see that the T cells are the amount of T cells are downregulated, while the B cells are the number of B cells are increased. That means the NPY can regulate the proliferation of this immune cells. So next, we performed single cell sequencing in spleen, and upon the downregulation of NPY in CG neurons, and we found that when we look down NPY in CG neurons, the proportion of different cell types in spleen was changed for. And we also found that the genes in the spleen were changed, especially the upregulation of immune response genes. That means upon the lockdown of NPY in CG neurons, the immune system was somehow activated. So which means NPY can generally act to inhibit the activity of spleen immune cells. And next we asked which cell type they regulated. So we performed based on our single cell sequencing, we checked the NPY receptors in different cell subtypes, and we found that NPY receptors are mainly expressed in T cells and the macrophages, as you can see here, but not in B cells. And then we isolated the T cell and the B cell and the macrophages from a spleen, and then we treat these isolated cells with NPY. What we found is that the B cells has no response to NPY, while T cells and the macrophages significantly respond to the treatment of NPY, which is in fit with the expression expression of NPY receptors in these cells. As you can see here, in T cells and macrophage cells, the NPY receptors are highly expressed, but in B cells there's no expression of NPY receptors. But in our RNA-seq data, we also see a response of B cells, but that may probably due to the indirect effect, because this experiments were performed in vitro, but the siRNA experiment, siRNA and single RNA sequencing data was performed in vivo. So the response of T cells and the macrophages can regulate B cells further. And we found that in the macrophage cells treated with NPY, the cytokines such as CCL2, CCL1, and IL-1 beta and the TNF alpha, those TNF alpha signaling pathways are downregulated. So that means NPY is directly acting on the T cells and the macrophage cells in spleen, and the TNF alpha signaling pathway might be one of the important cellular mechanism of NPY effect. Next, we performed an ex vivo experiment. We isolated only the spleen cells and the CG neurons, because it's very hard to exclude the HPX effect in vivo, because if we look down the NPY or if we treat the LPS, they may have a lot of very complicated reactions from different organs. So here we only isolated the neurons from the CG and the immunocells from the spleen, and we co-culture the isolated neuron and the spleen cells together. And then what we found is that if we look down NPY in this co-cultured neuron cells, so we only look down we firstly culture neuron cells, and we look down this neuron, look down NPY in these neurons, and then we co-culture the immune cells. So, and then we check the expression of this cytokine genes, like TNF alpha, IL-6, IL beta, and CCL2. What we found is that LPS induced a much higher level of TNF alpha, IL-6, and other cytokines when NPY genes was locked down in the cultured CG neurons. That means the neuron-derived NPY can indeed inhibit the immune response in immune cells. Next, we performed an in vivo experiment to prove that the NPY lockdown in CG neurons can indeed influence the defense to bacteria and LPS. So what we did here is we first looked down NPY in CG neurons, and then we infected them with LPS, or we use the bacteria for the infection. So what we found is that the inflammation cytokines was much higher in the NPY lockdown groups, and when we overexpress the NPY in the CG neurons, we observe the opposite effect. So and we also treat the rat with NPY antagonist, and we found that the NPY antagonist treatment can increase the LPS evoked inflammation which suggests that the NPY acted through the NPY1 receptor to inhibit the inflammation. And we also performed another experiment to prove that this phenomenon is indeed through the regulation of neural system. So what we did is that we perform a denervation experiment by by 100% ethanol. So with that, we can destroy the connection between the CG neuron and the spleen. And then we look down this NPY in the spleen again, in the CG neuron again. Sorry. So as you can see here in the control group, the lockdown of NPY in CG neuron can indeed upregulate the inflammation. However, if we disconnect the crosstalk between the nervous system and the immune system, this upregulation effect was gone. So next, as NPY is a very conserved neurotransmitters. Next, we asked whether NPY or NPF is a conserved language for the crosstalk between nervous system and the immune system. So here we use the Drosophila as another animal model. So we looked down the neural NPF in the flies, and then we performed the infection, and what we found is exactly the same like what we found in rat. So lockdown of NPF can indeed promote the inflammation, and the more flies die because of this inflammation storm. So this is that means this pathway is conserved between different animals. So next we checked whether this is also the same story in human. So we collected a lot of samples in human and from the autoimmune disease patient. And what we found is that so firstly we tried that we isolated the immune cells from a human blood and treat them with ARPS. We found that they are upregulated upon the treatment of ARPS, and then we used we added the antagonist of NPY. We added the NPY there to inhibit the immune response. And what we found is that NPY can indeed reduce the ARPS induced expression of the cytokines in human immune cells. That means the NPY have the same function in human cells. And we also found that we also used some public GWAS data. We found that the NPY and NPY receptors mutation can indeed induce a lot of a different kind of autoimmune disease. That means NPY or NPY receptors, those pathways are involved in autoimmune disease, suggesting that NPY is the regulation of immune systems by NPY pathway is also conserved in a human and may involve in autoimmune disease. So therefore, we collected the serum samples from the autoimmune disease patient, and what we found is that the NPY level in the serum is indeed significantly lower in the patient, we tried two different kind of autoimmune disease model, the RA patient and the SLE patient. Okay. And then because we cannot perform the siRNA in human in human, so we come back to the animal model. So we firstly knock down NPY in mice, and then we make a autoimmune disease model in mice. And what we found is that we use the CIA model and autoimmune disease model. Okay, what we found is that when we look down NPY in the CG neurons, we found that we found a more severe phenotype of this autoimmune disease, and we see more expression of this cytokine genes in this mouse model. So that means the NPY in CG neurons can indeed involved in the autoimmune disease. Okay, so the take-home message of this story is that NPY or NPF is a very old language for the crosstalk between nervous systems and the immune systems in different species. And the neuronal NPY is dramatically increased upon infection, and then the NPY is released to the immune systems, and then fine-tune the immune balance during inflammation response, and also autoimmune disease. So this story was published 2 months ago, and then we are very happy that the editor read comments in Nature Review Neuroscience, and they said they drew a very beautiful cartoon that the helicopters can perform the firefighting to dump the inflammation response to RPS. So as people often introduce some unpublished data, so I also prepared 3 minutes slides for some unpublished data here. So as people know that in our group, we focus on 2 fields. One is the neuroimmunoregulation system biology. Another one is developing a single cell spatial omics technique. So actually 50% of our people are working on technical development. So here I would give a very short introduction about the 2 papers we are submitting now. So the first one is called PyFish. It's kind of upregulation of iron scope. You can imagine in this way. So we can perform by this technique, we can perform multiplex fish in vivo and in different species. As you can see here, we performed a 9 gene hybridization in S1 cortex. So this is the real picture of the hybridization, and this is pictures after the decoding. As you can see, we can see clearly distribution of this layer-specific genes in the cortex from layer 2, layer 3, layer 4, layer 5A, layer 5B, and layer 6. And then we can wash this gene away and then perform another round of fish. So we can get totally 21 genes by 2 rounds of hybridization, as you can see here. And then we can perform the integrated analysis of this this fish data and single cell data. So we can, with that, we can annotate the single cell sequence in a spatial level. That means we can delineate the architecture of our cell type in the brain. And but that is a still low throughput. We can or middle throughput. We can get maximum 20 or 30 genes by this fish technique. So next we I don't know how to move this. Okay. So we are working on this spatial omics from 9 years ago. So but we are submitting these papers. So basically there are three techniques for spatial omics: the NGS based, the fish based, and the in situ sequence based. So we are working on the in situ sequence based spatial omics. I don't have time to explain or to introduce the advantage and the disadvantage of those different methods, but maybe next time, if we get more time, we can introduce more about this. So so we developed a single cell, single base pair in situ pairwise in situ sequence technique called MIPSEQ. And okay. As you can see here, by 3 rounds of in situ sequencing, we can get the in situ expression and also the sequence information of 247 genes in hypothalamus. As you can see here in the second round. So this is a raw picture of the second round sequencing or imaging. With that, we can get a spatial cell type map, and then we can also see the pseudo trajectory map of this cells in spatial level, and we can also see the spatial cell communication in the brain. So we know that by single cell sequencing, you can get a cell interaction map, but that has no spatial information. Here we have with this spatial information, we can say the spatial cell communication. So, and we can also do this experiment by at different level. We can detect a protein, RNA, and DNA at a spatial level. So we call it spatial multiomics. To visualize the localization of the proteins, we use a strategy that in which we conjugate the DNA to antibodies. So the antibody can specifically recognize the proteins, and then we can we switched the immunohistochemistry to in situ sequencing by this conjugated the DNA. With this technique, we can simultaneously detect the expression of different genes and the protein and also the DNA. We can also combine the 2-photon imaging with this spatial multiomics data. So we firstly perform the 2-photon imaging, and then we cut the brain and perform the in situ sequencing, and we align this in situ sequence pictures with the 2-photon pictures. So with this, we can see the dynamic. We can see the we can integrally analyze the different level of informations, like the function or the calcium dynamics of these cells and the expression profile of the cells. For example, we can see different kind of calcium dynamics here, and we can also see the expression profile here. So we are collecting more and more the cells, and hopefully we will get more resource, and then we can use artificial. We can use deep learning to predict the custom dynamic by the expression profile and vice versa. We can also predict the gene expression by the custom dynamic or by the function. So with that, I would like to thank all the people involved in this work. And we get a lot of good suggestions from Professor Qiufu Ma. This work are many performed by two very excellent student, Xiao Ke and Jin Song. We also get a lot of help from Professor Ji Hu and Professor Zheng Guo and Yun Han. So I'm sorry I was kind of late this morning because of the Zoom system of my computer. I changed 3 computers and finally it worked. Okay, thank you.
Welcome. Thank you, Gang. Just beautiful, amazing talk. I have some questions, but I'll move to the question section. So
okay.
I would like a quick one. I have a ton of questions I'll ask later, but first one and the only one I'm asking for now is, so would you suggest some automatic system regulating the NPY releasing from the central nervous system somewhere? So I'm curious about where the source of the NPY regulating from the brain, right? How we gonna release different kinds of NPY so we can resist the bacterial infection? Do you know how the NPY is released by the brain, by automatic system or some other system, by motion even?
I think it's in this case. I think it's the NPY is synthesized in the CG ganglia, in the ganglia, but not in the brain.
Okay.
So the CG neurons can directly sense the infection of the pathogen because they express the LPS receptor like Tor-like receptor 4. I think they also express other cytokine and receptors, so they can directly sense the invasion of different pathogens.
Okay. Yeah. So will it be regulated by brain or not? You don't know yet.
I think they can also be regulated by the brain because we are working on another project. We want to investigate how the brain can influence this ganglia. For example, in depression, anxiety and stress conditions.
Okay, thanks. All right, any more questions?
So there's one in the chat.
Okay, here's Gang said chat from a question for Daisy. You can ask it yourself if you want.
Okay, I will check.
Yeah, for how do you fix the region exactly after washing the previous colors in the in situ?
Okay. So we used different this kind of detergent or this kind of stuff, and we increase the temperature. Then we can wash the first hybridized probes. So after that, we can perform the second round of hybridization or the second round of in situ sequencing, because hybridization probes are very short. So if we increase the temperature, they will be released. But we include some detergent.
All right, any more questions? Yeah, I'm curious about whether be the next RNA scope gonna use. So how do you compare RNA scope for your technology?
Okay, so I didn't show the data here, but actually we indeed compared the RNA-scope probes with our PyFish probes. We are slightly better, not significantly, but slightly better. But we
you can license out your technology.
But we are still optimize this. We are still optimize this probes. We want to make them very robust and stable.
Okay. Any more questions? Okay. So yeah, great. So we're a little bit over time for now, but talk is great. Thanks so much for coming. I'm gonna thank Renata and Gang for their beautiful talks. Thank you for coming. Aaron, thank you. It's great.
I'm really sorry. I was.
No. It's great. The talks mix up. Okay.
Okay.
Really appreciate it. Really enjoy it.
Yeah.
Thanks Renata. Thanks Gang. Thanks everybody for coming. See you all next week. See you around.
Okay.